Dear students, today we shall learn an important topic, that is sodium potassium ions pump. So before going to this one, we need some introduction. Say for example, you must understand what is gradient, how many types we have. Then next aspect is phosphorylation, and dephosphorylation. So these two concepts must be understood for understanding this sodium potassium ions pump. Now first of all, let me give the meaning of this gradient. Gradient means it is nothing but a gradual change. A gradual change. So this change may be either increased or decreased. So this constitutes a gradient. In physiology, so you are all biology students, so you can understand what is a physiology. We have separately plant physiology, animal physiology, like that. So physiology means actually it concerns with the chemical processes or biological processes that are occurring in both plants and animals. Now in physiology, we have two types of gradient. One is concentration gradient. Concentration gradient. And the second one is potential gradient. Potential gradient. In the case of human beings, so we are studying this sodium potassium ions pump with respect to human beings itself. So now these ions, sodium or potassium ions, we can express its concentration, whether it is having high concentration or low concentration or moderate concentration, like that. So sodium and potassium, it can be expressed either in concentration, or we can also express these ions with respect to charge also. Sodium, it is having 1 plus, and potassium is also having 1 unit of positive charge. So that means its information can also be given in the form of its charge. So we are speaking about the same matter. Matter is same. Ions are sodium, potassium, we may have magnesium ions, calcium ions, whatever may be. So that comes under matter. So this constitutes matter. So this matter, its information is expressed either in the form of concentration or in the form of charge. So if it is expressed in the form of charge, we use the word potential. Concentration is concentration itself. So gradient is classified into two categories, that is a concentration gradient and a potential gradient. Why this concentration gradient will arise? At one place it is having high concentration, at another place it is having low concentration. Or similarly, at one place it is having a high charge, and at other place it may have few ions, so the charge will be low. So like that here, concentration gradient or potential gradient, it is mainly due to unequal distribution. Unequal distribution of ions. So if we speak it in terms of concentration gradient, at one place it is having a more amount of these ions, so its concentration is high. At another place, the ions number is less, so its concentration is low. So because of this one, what happens? We are considering about processes occurring in biological systems. That means physiology. We are studying the physiology. From which area to which area these ions are moving. Generally, they will move from higher concentration to lower concentration. So, this concentration gradient, it is mainly due to unequal distribution of ions. Why potential gradient is occurring? That is also due to unequal distribution of charges. Okay. Now, I will give another important term regarding this one. Transport phenomena. Transport phenomena. Transport means exchange. So we are moving ions from one place to another place. Generally, this transport phenomenon, it is classified into two categories. One is inactive transport phenomenon and another one is active transport phenomenon. What is this inactive transport phenomenon? Now in this, generally from high concentration to low concentration, the ions will move from higher concentration to low concentration. This is a natural process, that means it is a spontaneous. It is called spontaneous process. For this to happen, no need of energy. There is no need of energy in the case of a spontaneous process. Why? Because it is occurring naturally. Why? Because high concentration area is there and a low concentration area, and there is a passage between these two. There is a connection. So that's why ions will move from higher concentration to the lower concentration. I will add one more word here. It is moving along the concentration gradient. We write like this. So, with respect to concentration gradient, ions are moving. Naturally, they are moving. Spontaneous processes. So, for this, it requires no energy. Such a transport phenomenon is called inactive transport phenomenon. Now, come to the second one. Active transport phenomenon means, here in this case, from low concentration, the ions are moving towards a high concentration. So this is not a natural process. So it is moving. So ions are moving against the concentration gradient. Against the concentration gradient, the ions are moving. Natural process is from higher level to lower level. Higher concentration to lower concentration, ions must move. But here it is moving from already low concentration area. From here we are shifting, we are transporting, we are exchanging the ions from low concentration to higher concentration. For this, it requires some amount of energy. Energy must be required. Such a transport phenomenon is called as the active transport phenomenon. For your easy understanding, I will give one example. A familiar example. So, for example, here we have a wooden plank. So this is ground. Now, a boy or a girl, a child, is sliding from top to bottom on a wooden plank. This is a wooden plank. So, along the direction of gravitational force, the body will move. So here in this case, no need of energy for moving this body from a higher level to the lower level. It requires no energy. So this process is called inactive form. Suppose if I want to move from bottom to top, that means it is against the, here we are moving the body against the gravitational force. That's why we must supply some amount of energy. Then this type of transportation is called active transportation. Okay, so that you must remember. So transport phenomenon means we are exchanging matter and energy from one region to another region. So this sodium potassium ions pump, it is an example for active transport phenomenon. Generally, in the case of human beings, that energy will be in the form of ATP, adenosine triphosphates. Energy is required for moving ions from an area of low concentration to higher concentration. That is called as the active transport phenomenon. Is it clear with this word? So sodium potassium ions pump, it is an example for active transport phenomenon. Next I am moving to another one. Generally, so these sodium potassium ions, active transport phenomenon, in this case, energy is nothing but ATP we are using. Generally, which proteins? The proteins means it is a combination of amino acids. So, which amino acids generally involved in active transport system? First one is serine, C-E-R-I-N. This is one amino acid. A group of amino acids constitutes a protein. Serine, then threonine, tyrosine, then last and final, histidine. So these are the amino acids generally which are involved in the active transport system of whether they sodium or potassium ions or calcium ions, magnesium ions, whatever. So now I am taking this serine as an example, and we shall move to second important term, that is what is phosphorylation and what is dephosphorylation. Phosphorylation means addition of phosphate group. Addition of phosphate group, generally PO4 minus 3. This is phosphate group. PO3 minus 3, phosphite. So now here we need only phosphate group. So addition of phosphate group is called phosphorylation. Now another one what I told, dephosphorylation. Dephosphorylation. This is removal of phosphate group. So removal of phosphate group is called as the dephosphorylation. Now I am taking serine as an example, and let me explain that process. These are the two processes. Serine. So serine, serine, S-E-R-I-N. It is an amino acid. Amino means it must have amino group, and it is having a carboxylic acid group. So amino acid. Now its valence is one valence with hydrogen, second, third, there is one more. So CH2OH. This is the formula of serine amino acid. Now, for convenience, I am writing this OH here, like this. Now, this is serine. Now, this H is removed. As a result, here I will write H positive. Now, O will become minus. Now, here this is an active transport phenomenon. Active transport phenomenon means it requires energy. Generally, in living organisms, the energy that is utilized will be adenosine triphosphate. So, it is an ATP. So, here we have ATP that will split up and forms ADP, adenosine diphosphate, plus PI. This is very important. What this PI stands for? I stands for inorganic. P stands for phosphate group. So inorganic phosphate plus energy. So energy is also released from this ATP that is used for sending these ions. If we take actually the process, then you can easily understand that. For the time being, just try to understand what is the meaning of this phosphorylation, and the next one is dephosphorylation. So here we got, when once this H is removed, then what is the left over here? Then H2, then CH, then COOH, we have CH2, then O is there. To this O, this inorganic phosphate will attach. So this is phosphorus 1, 2, 3. So totally PO4 minus 2. This is a phosphate. Now its name is serine phosphate. So this is a serine phosphate. So in the phosphorylation process, what is happening? Addition of phosphate group. Where it is going, where it is being attached to the amino acid, it is attached. That process is called as the phosphorylation. Addition of phosphate group to the amino acid or a group of amino acids is called as a protein. So to the protein, this inorganic phosphate group is being attached. That the process constitutes phosphorylation. Now I am moving to the reverse situation. Now one more point I have forgotten here. Generally, phosphorylation in the case of human biology, physiology, generally protein kinase. This is the enzyme that is catalyzing this phosphorylation process. You must remember this one also, protein kinase. This enzyme is involved in this process. Now I am moving to the second one, dephosphorylation, opposite. That means here I will write directly like this. Once again it is given. So removal of addition of phosphate group is phosphorylation. Dephosphorylation means removal of phosphate group. So here I will write minus PI. Phosphate group is removed. As a result, what happens? Once again this H, it will combine with this O, and we are getting once again serine. So this is dephosphorylation. Here no need of energy. Here it requires no energy. That means generally this reaction we can write it like this: ADP and phosphate group is once again coming, and if we add some amount of energy, then we can get once again ADP. So once the energy is utilized, it cannot be regained once again. So In order to balance this equation, I am writing like this. So if ADP means adenosine diphosphate and inorganic phosphate is detached from the amino acid, and if we supply energy, then we are getting ADP once again. So this process is called as dephosphorylation. So this is dephosphorylation. This is catalyzed by another enzyme. Its name is protein phosphatase. TAS. Generally, enzymes name ends with EAS. Kinase, phosphatase, or this can also be catalyzed by protein synthase. That is another enzyme. So this is dephosphorylation. So in the case of phosphorylation and dephosphorylation, what is happening in the first process? Phosphate group is being added. In the dephosphorylation, phosphate group is removed. Now the best example here is sodium potassium ions pump. Now I am entering into the sodium potassium ions pump. How it works and what are its applications. Why it is very important in our body. Let us understand its applications first. Then I will move to the mechanism. In the applications part, first one is, if sodium ions and potassium ions are present in a balanced condition, throughout the body, then what happens? This facility, kidneys, to filter waste product that is present in the blood effectively. So it will effectively filter the waste materials that are present in the blood. And secondly, it reabsorbs if glucose is present in the blood. Suppose if the filtration is not good, then glucose will also be coming out into the urine. So that's why kidneys, if they want to work effectively, then throughout the body we must maintain a correct balance of sodium and potassium ions in the throughout the body. So second point, in the first point itself, second aspect is reabsorption of glucose that is present in the blood. And another one is maintenance of proper pH of blood. Blood pH will be normally human pH is 7.35 to 7.45. Generally 7 is neutral, 7 to 14 basic, 7 to 0 or 1 it is acidic. So our blood is slightly basic, greater than 7. So 7.35 to 7.45. So in the human body, pH must be properly maintained, and another one is electrolytes level. Electrolytes level in the blood must also be maintained. Electrolyte means any substance that may be an atom or a molecule or ion which which allows the electricity to flow through it, or it will move from one end to another end by carrying the charge. Electro means charge possessing ions generally. So electrolytes level means not only sodium and potassium, other element ions are also be taken as electrolytes. So in the first point we are writing that there is a balance between sodium and potassium ions throughout the body. This is must, it is necessary. Why? Because by which only kidneys will filter the waste products that are present in the blood. They will filter effectively and they will reabsorb. Generally, kidney's smallest part is called nephron. It is having bowels, capsule. So filtration process is going on there itself. So the nephrons, they will absorb the glucose that is present in the blood. Then they will maintain. So this ion balance is maintaining a proper pH of blood as well as electrolytes level. This is first aspect. Now I am moving to the second point in the application of this sodium potassium pump. It will control BP, blood pressure. BP is controlled by these ions. So if the ions number, if it is increased, if it is not balanced, then if it is in excess, then BP will increase. So blood pressure is under the control of these ions. Then next one, myocytes. Myocytes, calcium ion balance. And cardiac, contractility. What are myocytes? Myocytes means the special type of cells connected with tissues. Generally, its function is in order to contract or to expand. Say for example, if we take the heart will contract. That process is called systole. If it expands, then it is called as a diastole. So here the muscles, the cells that are present in the heart, they must contract and expand. For doing that function, we need these myocytes. And these myocytes, they will have a proper balance of calcium ions. This is required for doing that one, that is systole and diastole process. So in the second point what we are writing, the sodium potassium ions, it is controlling the blood pressure and it is controlling myocytes, calcium ion balance and cardiac contractility. So contraction means systole. So cardiac contractility and myocytes calcium ion balance and control over blood pressure. This is the second important function of sodium potassium ions pump. Now I am moving to the third one. Impulse and stimulus. In the case of nerve cell is called neuron. So neuron will have dendrites, axon. So I am not going to the details of structure of nerve cell. So in the case of nerves, the information is passing from central nervous system to body. And from body also information is moving through the nerves and it is reaching the central nervous system. Suppose here, let us take a small needle and if we hit it on a finger, then that constitutes stimulus. That means here information will move from body to central nervous system. Then in the central nervous system, it will observe what is that is happening to that forefinger. Then it will give the information. You just remove it from that needle. So impulse, that is called as the impulse. From central nervous system to body parts, the information is coming. So if the proper balance of sodium and potassium ions are there in our body, then the impulse to response will occur effectively. In order to have impulse to stimulus response, impulse to stimulus, impulse is also called response. So impulse to stimulus, this process, if it has to be done perfectly, then we must maintain its balance. Sodium and potassium ions balance is a must in the body. Next, fourth one. It controls or regulates cell volume. What should be the size? What should be the volume of a cell? This is under the control of these ions. Suppose if the sodium potassium ion pump, if it is dysfunctional, if it is not working, then what happens? The cell will get swollen. So, wherever the cell is undergoing swollen, means it there sodium potassium ion pump, it is not functional. So this is the fourth application of this. Last and final, for overall, so it is not visible, I think. Here, right? So fifth point is overall health of cells. So cells constitutes tissues. So if the tissues are, if they want to work perfectly, then ions concentration must be maintained properly. There should be a balance between these ions. So this is the application part of sodium and potassium ions pump. Now I am moving to the exact function of the structure and function, how it is happening, phosphorylation and dephosphorylation, how it is happening, that we will learn. Yes. Sodium potassium ions pump. This was given by Christian Scope. So Gens, GENS, Christian SKOU Scope. He has given a satisfactory explanation how ions get into or out of cell. How ions are entering into the cell and how ions are leaving the cell, moving outside the cell. He has given a satisfactory explanation and a mechanism also. That's why he has received a Nobel Prize also. So germs Christian score. Okay. Now I am coming to this structural aspects. First one. All animal cells without any exception. So in the plasma membrane, we have this sodium potassium ions protein. So we write like this also: Na positive, K plus, ATP is. That is the another way of writing this one. This is a protein. Protein means a group of amino acids. So cluster of amino acids that constitutes a protein. Our body itself is made up of proteins. So membrane, all animal cells membranes, in the cells we have sodium, potassium, ATP is protein. So we can call this one as a protein membrane. We can also call it as a protein membrane, or it can be called as sodium, potassium, ATPase. ATPase means it is an enzyme. All enzymes, they will have the ending ASE. Why here I have written ATP? Because sodium potassium ions pump, it is an example for active transport phenomenon. Active means it requires the energy. So in the biology, the energy resources are ATP, adenosine triphosphate. That's why here I have written sodium potassium ATPase, or we can say sodium potassium ions pump, or we can say protein membrane. They are one and the same. Okay. Now this protein membrane, sodium potassium protein membrane, it is having two units. It is having two units. One is a large alpha unit and a small beta unit. In the alpha unit, it is having three sites. Three places are there, vacant sites. And beta unit is having two sites. What for? For accommodating ions. So alpha unit is bigger in size, it will accommodate 3 ions. Beta unit is smaller in size, it is having only 2 sides, so it can accommodate only 2 ions. Okay. Now I am drawing the diagram also simultaneously. Just observe please, all of you. Okay. Now draw lines. So this is small in size, whereas this one is large in size. This is alpha unit and this one is beta unit. It is having 3 sites for accommodating ions, and here we have 2 sites only for accommodation of these ions. Now this is called opening. Okay. Now here I am drawing dotted lines. this stands for? This is intracellular fluid. This is intracellular fluid. At the top we have this is extracellular fluid. So this total constitutes plasma membrane. It is a plasma membrane. So in this we have sodium, potassium, ATP, ase enzyme in the membrane itself. Now this is the opening. In the case of intracellular fluid, in this we have sodium ions and potassium ions are also present here in the intracellular fluid. In the extracellular fluid also sodium and potassium ions are present. Now this one, sodium potassium ions pump, it is an example for active transport phenomena. Active means energy must be spent, energy must be utilized. So here that means ions will move from low concentration to high. That means in the intracellular region fluid, sodium ions concentration is low. From low concentration, it is moving towards the higher concentration. That's why it is called as active transport phenomenon. Okay, so this much you must remember. Now, through the opening, sodium ions, they will enter through this opening. They will enter into the alpha unit and they will occupy these places. So here we have sodium ion, and three sodium ions. Draw another one, parallel line. This is alpha and this one is beta. So here they are entering like this. So they have reached Na positive, Na positive, and so in the first diagram, no need to mention these things. We just write sodium ions, they are moving like this. They will enter like this. And they are occupying these three states. Now this process is called phosphorylation. Phosphorylation. What is phosphorylation? Addition of inorganic phosphates. So here, let us take ATP. It will be converted into ADP plus PI, inorganic phosphate, and energy is released. So ATP is utilized and it is converted into adenosine diphosphate, then inorganic phosphate, and energy is released. So this energy is utilized for moving these ions from low concentration area to higher concentration area. Are you catching the point, please? So now they will fix it into the sites that are present in the alpha unit. And this inorganic phosphate, it gets attached to beta unit. So this is PI. And from where we get that inorganic phosphate? From ATP. ATP will disintegrate and it is releasing energy and it is converted into adenosine diphosphate and inorganic phosphate. That phosphate will attach to this one. Then what happens? If this phosphate, inorganic phosphate is being attached to the beta unit, then it will close that opening. So this is the opening we have. So we have to draw intracellular fluid must be, and here extracellular fluid. Okay. Now the second diagram. So here this is phosphorylation. This is catalyzed by which enzyme, please? Protein kinase. Protein kinase. And now in the next diagram, I will draw that one first. So this is extracellular fluid, and here we have intracellular fluid. This is intracellular open, and okay, here we have sodium plus, Na plus, Na plus. So these, once this Pi gets attached to this one, what happens is the opening will be closed here. You have to imagine like this. Now it is present in this position. When Pi gets attached to the beta unit, then what happens? Opening that is present at the intracellular fluid, it gets closed and opens at the extracellular fluid region. So we must reverse this diagram. Here it is inverted V. Now we have to draw V. Normal V. Okay. Now opening is there at the top, that is the extracellular fluid region, and in the intracellular fluid region the opening gets closed like this. Are you following this? So here also I have to draw that. Now here sodium ions are there, Na plus, and Na plus. These ions will escape into extracellular fluid like this. Is it clear? Now at the same time in the phosphorylation, what happens is potassium ions, its concentration is low. So in the intracellular fluid, potassium ions concentration is high. So from low to high, they have to be transported. That means that is also coming under active transport phenomenon. So at the same time, what happens? When the sodium ions are escaping, then at the same time potassium ions they will enter into the beta units. So now I will change this diagram. We have to draw this actual V shape. So this is beta, and this is beta, and this is O. And sodium ions they have escaped, and we have here potassium ions. So this is extracellular fluid, and this one is intracellular fluid. And beta is having inorganic phosphate. So Pi, Now what happens in the next process? Pi removed. So as a result, what happens? Once this Pi gets detached, then the closed end at the intracellular fluid region that will get opened, and at the opening that is present at the extracellular fluid region that will get closed. So in the next diagram we have to draw like this. Reverse. First one. This is beta. So this is alpha and beta units. This is completely closed, and this is extracellular fluid, and this is intracellular fluid, and there is a opening here. Now, why? Because inorganic phosphate is removed. So potassium ions that are present here, they will enter into intracellular fluid. At the same time, what happens? Sodium ions, once again, they will start moving into their alpha unit sites. So this is a dephosphorylation. So in the dephosphorylation, what is happening here? Dephosphorylation, here we can write ADP plus PRE. If we supply energy also, then once again we get ATP. So inorganic phosphate is detached from this beta unit, so it will mix with ADP. Then we cannot get ATP provided if energy is supplied. So write like that only. If energy is given, then only we get ATP. Now this process is called as the dephosphorylation. Dephosphorylation. Now this is catalyzed by protein phosphatase, or protein, another name is the protein synthase. Okay. So this process constitutes dephosphorylation catalyzed by either protein phosphatase or protein synthase. So general meaning of synthase is energy is not required. So this is a inactive process here. Now energy is not spent in the dephosphorylation, whereas in the case of phosphorylation we need energy supply. So this constitutes sodium potassium ions pump. So it is an active transport phenomenon. Why? Because we are moving sodium ions from low concentration to high concentration, and potassium ions from low concentration to high concentration only. That's why it is going against the concentration gradient. The concentration gradient to viruddhanga jarugutandi process. Generally, ions must move from higher concentration to low concentration, but it is moving against this concentration gradient. That is, from low to high it is being transported or exchanged. That's why that transport phenomenon is called as the active transport phenomenon. Okay, this is very, very important. Actually, it is an essay question, but it may appear as a short answer also. Thank you.